When a pathogen breaches physical barriers, your body faces a strict deadline. It must launch a lethal response within minutes to prevent a systemic infection. But constructing a highly specific, lifelong defense takes days. The immediate reaction falls to the innate immune system. It is nonspecific, always active, and treats a cut from a rusty nail the same way it treats a viral droplet. Crucially, it is entirely incapable of forming immunological memory. The adaptive immune system operates on a completely different timeline. It requires delayed activation to generate a targeted attack against a highly specific pathogenic antigen, but it leaves behind a lifelong cellular memory of that exact invader. This creates a mechanical problem. Because the adaptive system is locked away in the lymph nodes, it is completely blind to the initial breach. There has to be a physical mechanism to precisely transfer pathogenic data from the innate first responders on the front lines back to the adaptive lymphocytes. Mastering immunology for the MCAT requires looking past the individual cells to understand the exact molecular handoffs between these two operational timelines. That data transfer happens across a biochemical bridge known as the major histocompatibility complex, or MHC. Both branches of the immune system share a common cellular origin. They all differentiate from hematopoietic stem cells in the bone marrow. The innate granulocytes and the adaptive lymphocytes are distinct tools manufactured from the same source material. Once developed, specific innate sentinels, like macrophages, take up permanent residence in the body's tissues. They act as the immediate first responders to local physical breaches. As these sentinels consume and destroy the immediate threat, they perform a secondary, vital role. They harvest tiny peptide fragments from the destroyed invader. These fragments are the antigens. The biological source of those harvested antigens dictates exactly which immune cascade is triggered next. The immune system divides all incoming threats into two distinct MHC pathways. The first pathway handles extracellular threats, like free-floating bacteria in a wound. These trigger the exogenous antigen presentation pathway. When a macrophage phagocytizes a bacterium, it traps the invader in an internal vesicle, degrading it enzymatically. Specific bacterial fragments are loaded onto an MHC2 molecule. This complex travels to the outer membrane, displaying the antigen. MHC2 exclusively presents exogenous antigens to CD4+ T cells. This specificity exists because extracellular threats require a systemic humoral response, meaning antibodies circulating in the blood. CD4+ helper T cells are the essential orchestrators that recruit those specific defenses. Intracellular pathogens require a completely different response. Viruses bypass innate phagocytes entirely and infect host cells directly, turning them into viral replication factories. To counter this, the body uses the endogenous pathway. All nucleated cells constantly degrade internal proteins, including viral ones, loading them onto an MHC1 molecule in the endoplasmic reticulum. The loaded complex migrates to the cell surface as an external audit. This strict rule means MHC1 presents endogenous antigens exclusively to CD8+ cytotoxic T cells, which then execute the infected host cell. Some viruses attempt to evade this by downregulating host MHC1 expression. To close this loophole, natural killer cells patrol independently, hunting specifically for cells lacking these markers and inducing apoptosis when they find them. A common mnemonic to cement this logic is the rule of 8. CD4 multiplied by MHC2 equals 8. CD8 multiplied by MHC1 equals 8. The system perfectly matches the location of the threat to the correct cellular executioner. Returning to our CD4+ helper T cells, their activation triggers a cascade that initiates the humoral response. This drives B-cell clonal selection. Out of millions of naive B cells waiting in the lymph nodes, only the specific cell with a receptor matching the exact pathogenic antigen is activated to proliferate. This proliferation yields two distinct lineages. Plasma cells immediately begin manufacturing high volumes of targeted antibodies, while memory cells go dormant. The circulating antibodies released by the plasma cells neutralize the threat through three mechanisms: opsonization, which tags pathogens for destruction, agglutination, which clumps them together, and neutralization, which blocks tissue invasion. The ultimate goal of this entire architecture is the generation of those dormant memory cells. They guarantee that if the exact same antigen enters again, the adaptive system can bypass the innate delay and launch an immediate, overwhelming response. Theoretical models are only the baseline. High-level scientific reasoning requires applying this architecture to experimental data. To do that, we can look at a study designed to isolate the causes of immune-mediated liver failure during acute viral hepatitis. The pathology here is complex. Tissue damage is driven by multiple overlapping immune pathways. Researchers suspected 3 specific variables were driving the destruction: Phas-L, TNF, and perforin 1. To test this, researchers immunized mice with an OVA peptide to build immune memory. 30 days later, they initiated a secondary challenge using an adenovirus expressing that same OVA peptide. They quantified the resulting liver destruction by measuring 2 dependent variables: serum ALT levels, a primary clinical marker for liver damage, and the percentage of total body weight lost. To identify the true molecular culprit, the experimental strategy required systematically isolating and neutralizing each suspected variable during the secondary viral challenge. The first experiment tested Phas-L and TNF. Researchers established independent variables by pre-injecting the mice with neutralizing antibodies against either Phas-L or TNF, alongside an IgG control group. The physiological intent was to bind and disable Phas-L and TNF in the bloodstream, entirely preventing them from exerting their normal biological function. These 2 line graphs track the data over 3 days. The left graph plots serum ALT levels, and the right plots body weight loss. Look closely, all 3 lines cluster together, and the vertical error bars overlap significantly. Because disabling Phas-L and TNF produced no statistical deviation from the control group, neither factor is the primary requirement for immune-mediated liver damage. The second experiment shifted focus to the final variable, perforin 1. Instead of using neutralizing antibodies, the researchers changed their methodology. They transferred OVA-specific T cells into naive mice before hitting them with the viral challenge. This created 2 distinct test groups: mice receiving wild-type T cells and mice receiving perforin 1 knockout T cells. These line graphs present the cell transfer data. The wild-type group shows a drastic spike in serum ALT and a steep drop in body weight. However, the perforin 1 knockout group exhibits virtually no elevation in serum ALT and loses significantly less weight. We can connect this empirical data directly back to our theoretical model. Because this is an intracellular viral infection, it triggers the endogenous MHC1 pathway, which strictly activates CD8+ cytotoxic T cells. The experimental data proves that these CD8+ T cells mediate acute liver tissue destruction, specifically through the targeted release of perforin 1. True mastery of the material requires exactly this: